everyone. This video is to walk you through the Macromolecule Lab. So make sure you pull up the procedure from Socrates and you read all the way through the directions before you get started. Starting with part A, we're going to be talking about proteins. So we're going to be walking through some of these different macromolecules. Again, carbs, proteins, lipids, and nucleic acids. And we're going to be exploring a little bit more about how they connect and form and what exactly they mean. All right. Now your directions say that you can actually do these different parts separately from each other, but when you do start one part, make sure you have enough time to finish it. This lab should take you, about an hour and a half maybe, so you might just want to do it all in one chunk. Part A, starting with proteins, you should have some pipe cleaners in your lab kit, so you want to have those handy. And then the second part of this, you're actually going to need an egg, as well as your stovetop and a pot with some water. So starting with number 1 in part A, it says that we want to model a protein using our pipe cleaners. If you don't have different colors of pipe cleaners, you want at least 3 colors. So if you don't have different colors, you can take markers and color, maybe put dots or lines on them so you can tell them apart, okay? But we want a few different kinds or different colors if you have them. It says that you can also cut up some string or other material if you don't have pipe cleaners, but these should have been included in your lab kits. And each strand, so each individual pipe cleaner is going to represent the amino acid or our monomer of proteins, okay? So there's number 1. Remember, the amino acid is the building block or the monomer for our larger molecule, the protein. And a protein is a polymer, right? It's a big molecule made up of a lot of little amino acids. Number 2, it says get 6 strands with at least 3 different colors. Okay. And again, you can mark them if you need to, if you don't have 3 different colors. Each individual strand represents an amino acid. It says not all proteins have the same number of amino acids. Some can be shorter strands, some can be very long. But the sequence of the amino acids, the order that they're in, and how many of them will determine how the protein functions. So the functioning of a protein is all based on the ordering of your amino acids and the sequence, how long it is. That will then determine how it folds or what shape it takes, which will then determine what function it can do in the body. Okay, now we have tons of different types of proteins in the body. By now you should be able to list some different ones that we've talked about. And so think about those 20 to 21 different amino acids that you have to work with. So you have about 20 to 21 amino acid monomers, and you can build all different types of sequences to make your proteins. So you're going to make a protein. So it says write out directions for making your chain of amino acids. Now you have 6 different colors you're going to use. You can pick whatever ones you want. And if you have more than 3 colors, that's okay too. And you're just going to make a pattern with it, whatever you'd like. So in this box, write down what your pattern is going to be. Okay, so I have purple, green, and yellow. So I'm going to make a pattern out of these different colors. And so I will write down essentially the directions to do it. Okay. Number 3, it says you're actually going to make this chain now. So you've kind of talked about how you're going to do it, and now you're actually going to make it. You want these to be pretty strong. So when you bond 2 amino acids together, you actually form what's called a peptide bond. And peptide bonds are pretty strong. So you want to make sure that you twist these and get it pretty nice and tough. Okay. So I have my 2 individual amino acids. I have my lavender and my green. And then this twisted part here is going to represent my peptide bond, my connection. And it's really strong. It's very hard to break that if I pull on it. All right. So however you want to twist and connect, go ahead and do so. And you're going to be making that same model that you wrote up above. Okay. Now, once we connect all of these together, it will be what's called a polypeptide chain. Poly meaning many, peptide meaning those peptide bonds, and of course a big chain of them. All right. Now you could even have 2 different types of amino acids together. 2 different types together, or 2 of the same types together, right? So the sequence, again, will vary. Whether I put a purple one here, or if I put a green one, or if I put a yellow, it will change what the protein's able to do. So go ahead and pause the video, make your chain, and then we're going to look at number 4. So number 4, it says most proteins are made from more than one polypeptide chain. Okay, so this polypeptide chain is my first structure, my primary protein structure. All right, it's just a chain of different amino acids bonded together with peptide bonds. Now, some proteins, this is all it would be. It would be this polypeptide chain. It would fold in a very specific way, and then it would be a functioning protein. Okay. Remember when we talk about our protein forms, we have our primary form. Our secondary form would then either be, usually it's a coil or a folded sheet. So an alpha coil or a beta sheet. It's not the prettiest coil in the world, but you get the idea, right? Once it coils up like that, this would be considered my secondary form, okay? And then it could form another shape when it coils all together, and this would be my tertiary protein shape. When it's in my tertiary shape like this, it's now a functioning protein. Say it's an enzyme. This is my binding site maybe, where something is going to bind to it and start a chemical reaction. So the tertiary state, it is a functioning protein. When it's just in that long polypeptide chain, it's not going to be doing anything yet. It needs to first fold itself, right? And it'll either do that coil, or maybe it'll actually fold into a beta sheet for its secondary stage. Something like this. There's my secondary beta sheet folding. And then maybe it would take on that tertiary stage to be a functioning protein. Okay. So that's just with one polypeptide chain. Now, number 4, it says some only have one chain, but a lot actually have multiple chains. So I have my first chain here, and I'm going to go ahead and make a second chain. So get at least 4 more pipe cleaners and make yourself a second separate polypeptide chain and write down your directions. So for me, I would say I'm putting a green next to a green with a purple. I'll do another green and then a yellow. So I now have 2 separate primary stages, right? And these can actually combine to join and make one larger protein. In our lecture, our example of this was hemoglobin. Think of that protein that's in your blood. It's actually made up of 3 to 4 different tertiary proteins all bonded together. So here would be my secondary form. Okay, now number 5, it says gently fold your peptide chains over each other to make a 3-dimensional shape. Each place that your chains touch another chain, right? Each place where they touch each other, it represents a different type of bond. Now, peptide bonds are the bonds going between my individual amino acids, right? When I put these 2 strands together, they're going to be bonding with some other type of bond. They're not quite as strong usually, but those bonds could be a number of different things. It might be hydrogen bonds, it might be bonds between sulfur molecules. And so go ahead and gently connect your 2 peptide chains into a bigger 3-dimensional molecule. Now, as these 2 came together, they were making bonds that were not as strong as the original peptide bonds. So this actually is a bit more of a fragile state. Think of those hydrogen bonds. They're easy to break, right? They're not actually a real bond. They're just an association, an attraction. So this is where temperature can really affect our proteins, which we're going to see in the next part of this experiment. So we have some very strong bonds, and then we have some not so strong bonds that make up our proteins. Some of them are going to be easier to break than others. Keep that in mind as you go forward. Now it says, remember that temperature is essentially just a measure of the kinetic energy, or a measure of how fast those molecules are vibrating. Remember, all of the molecules around us are vibrating, whether it's a solid pipe cleaner or liquid water or the air we're breathing. And so temperature is a measurement of how fast they're going. So as you increase your temperature, you're increasing the movement of the particles in the substance. keeping that in mind, it then becomes clear that higher temperatures could affect some of the bonds, and maybe they're actually going to break some bonds as it heats up. Okay. Model higher temperatures on your protein by unfolding the peptide chains. But remember, our peptide bonds are quite strong. So what that means is I'm going to now take apart my two peptide chains. Okay. What you just did there, you heated your substance and you broke those hydrogen bonds, those sulfur bonds. All right. You can see the graph over on the side here. Now, as temperature increases, we can see that enzyme activity increases for a while, it peaks, and then once the temperature gets too high, that enzyme activity drops way off. Why might that be? So remember, enzymes are in fact a type of protein, very special and common types of protein we see in the body, helping out all sorts of chemical reactions. And so in order for a protein to function properly, it has to have its very specific shape. And when you break apart that shape, whether it's through heating it or another mechanism, you then essentially denature your protein. You ruin its shape, meaning it then cannot function the way that it's supposed to. So think about that with your graph. Think about enzyme activity dropping off after a certain temperature. Well, that's probably when you denatured your enzyme. Now down below it says humans have an average body temperature around 36 degrees Celsius. You guys might have heard in the news lately, I think it was actually in the spring that it was in the news a lot, that temperature for humans has actually been revised a little bit. It's actually a bit lower than we used to say, which is pretty interesting. That has to do a little bit with our evolution of our immune systems, how we have less inflammation in our body, how we're able to fight diseases a little bit easier. Okay, we don't have as much disease fighting going on at a given time, so our metabolism is a little bit lower than it used to be, which is a good thing. All right, it means that we're not fighting a bunch of parasites and pathogens like we might have been way back a couple hundred years ago. But roughly, we still say it's around 36 degrees Celsius. You could say the new norm now, I think, is around 97.5 degrees Fahrenheit instead of the 98.6 that it used to be. So it's dropped about a degree, scientists think. But that's just an interesting side note. So it says humans' bodies have an average body temperature around 36 degrees Celsius. How active are our enzymes at this temperature? So look up at the graph. Find 36 degrees on the horizontal x-axis and follow it straight up. Is it active? Not so active? Write down what you think. And then it says, what happens to enzyme activity if your temperature gets much above? Say you get a really high fever. What happens to those enzymes? And think about that in terms of why maybe fevers can be dangerous sometimes. And think about it in terms of why the body makes a fever at all. Why does our body want to heat up when we're sick? Is there any benefit to it? Okay, you've now finished with your lovely pipe cleaner polypeptide chains, so you can put those aside. And we're going to move on to the egg part of this. So you're going to want to get your safety goggles. You're going to need to go into your kitchen area with your stove. You want a pot and you want an egg. All right. Now to begin this experiment, you're actually going to have to separate the egg white out. If you've never done this before, there's a lot of YouTube videos and directions online. So just do a quick Google search. I personally like to just crack my egg in half and then take the two eggshells and you basically flip the yolk back and forth and let all the egg whites drop out into your pan. Maybe you drop them into a side bowl just so that if you're new to it, you don't accidentally drop the yolk into your pot and ruin your experiment. So maybe separate out your egg yolks into a bowl and then we can add them to the experiment once you know that the yolk is separated. Some people also crack the egg into their hand and just have a little space between your fingers and the egg whites will come through there as you hold and contain the yolk in your hand. So again, if you've never done it before, Google it. It's kind of fun to do. And you want the egg whites for this experiment, not the yolk. Okay. All right. Now number 6, it says put your goggles on, right? We're using hot boiling water, so you should have your goggles. The inside of the egg has two main parts. The egg white is called the albumin, and then it also has the yolk, okay? Now the yolk is very high in cholesterol and in fats, all sorts of nutrients for that baby chick, right? The white, the albumin, is very high in protein. So most of the protein in the egg is going to be the white part. Most of the lipid in the egg is going to be the yolk. And so that's why people who want lower calories tend to eat just egg whites. I don't think it's as delicious, but they do that because, remember, lipids have 9 kilocalories per gram. Well, proteins only have 4 kilocalories per gram, right? So you're cutting out a lot of the calories if you don't eat the yolk. You could be cutting out a lot of the nutrients too, though. So it just depends what you want. So for this, we are just thinking about the albumin, or the white part of the eggs. Now, I'm sure everyone's seen what happens to an egg when you cook it. But we're going to run that experiment in some boiling water, and you're really going to observe how that albumin changes in the water with temperature. So separate out your egg white, and we're going to put that egg white into some boiling water. You have a timetable here, so you can either use your phone or a stopwatch to keep track, but every 2 minutes you want to write down observations about the egg whites, okay? So when you're ready, at time 0, you're going to drop your egg whites into the pan of boiling water. You want your water to already be at a slow, rapid boil. Sorry, a slow boil, not a rapid boil. Just because if it's a rapid boil, it's going to really stir up your egg whites and it's going to be hard to really see it. So just a nice, soft, gentle boil is fine. Once your water is boiling, you can start time 0, drop your egg whites into the boiling water, and write down what it looks like. Right when you first drop the egg whites in, what is your observation for time 0? Okay. Now, keeping your heat nice and steady, again, not too high so it's not a rolling boil, but warm enough that it's close to a boil. Maybe you see some bubbles on the bottom of your pan. 2 minutes later, write down your description of the egg. You're going to go all the way up to about 14 minutes, or until your egg is completely white, okay? So when all of that albumin is white, then you can stop. Now, of course, again, everyone's seen cooked eggs, so you should have a pretty good hypothesis on what's going to happen. But again, write down detailed descriptions on your table at each time point, okay? You're looking at qualitative data. So what are the qualities? What does the texture look like? What does the color look like? What are the sizes of it? What exactly is happening? Now under your table, it says the egg white, or the albumin, protects the yolk and provides nutrition. Okay, again, this is an egg to turn into a baby chick, right? The eggs you buy in the store, by the way, are not fertilized, meaning it's just the egg from the female hen. There's no sperm from the rooster, okay? So there's no baby chick in there. But if it was fertilized, that is what it would become. So that egg, all those things that we love to eat from it, actually has really great nutrients for a baby chick that would be in there. Now, albumin is mostly just water and protein. So once you finish with this, after 14 minutes, you can go ahead and dispose of your egg and that boiling water, turn off your stove, and make sure you clean up your area, okay? Once you've done so, come back to the video, and we're going to look at number 7. So number 7, it says, describe what happened to that albumin. So you have a nice big table up above, summarize it. What happened? Remember how you unfolded your polypeptide chains of pipe cleaners? You did not, however, break those peptide bonds between the individual amino acids, right? So think about this with the albumin. What exactly did you do to the protein in the albumin? And it says below, this is an example of denaturing a protein. Denaturing means breaking apart the shape. You've changed the structure due to the heat, due to that extreme temperature, that extreme condition. But you didn't actually break the peptide bonds linking each amino acid together. The amino acids are still there, okay? So when you do eat a cooked egg, you're still eating those amino acids, and thus your body can then use them for its own purposes to build its own proteins, all right? Now, kind of an interesting note, if you guys don't know this, I've always kind of wondered why it turns white, Okay, so certainly you're breaking apart some protein structure, but why does it turn white? And it actually turns white because when you break apart those polypeptide chains, they kind of tangle up in ways that they're not normally tangled. They end up trapping water, right? Remember that albumin has both protein and water. So when you cook it, it unravels and denatures, it tangles it up and it traps water and it forms some new weird connections, and that actually turns it opaque. So it has to do a little bit with trapping that water and with the tangling of those peptide bonds after you've broken apart that initial structure. Kind of interesting. All right, so that ends our protein section. Now remember, as you think about proteins, keep in mind amino acids are the building blocks of proteins, and what the protein can do all depends on the sequence of the amino acids. That's going to be really important when we start talking about transcription and translation, which is the building of a protein, all right? So the order of those the order of those amino acids is actually dictated by our DNA. And so if we ever get mutations in our DNA, it's going to change the order of the amino acids in the protein, which could then make the protein not function in the way that it's supposed to. So keep that in mind for future lessons. Once you've cleaned up your station, we're going to jump down to part B and start talking about carbohydrates. Okay, so for part B, we're going to be talking carbohydrates. Now you need a couple different things. You need your iodine, which is that darker stain that you have in your lab kit. You should have your safety goggles for this as well. You want to have some paper around. Paper towels would be fine too. You should also have your Petri dishes, which are those shallow, circular, clear little plastic plates. And you want your pipette. And then you're also going to need a few different food items, and they can be food items of your choice. Now it says on here you want one from the grain category, so maybe some flour, maybe some rice or pasta, maybe cereal, bread, potato, okay? Your category 2 is going to be more of your fresher fruits and vegetables like cauliflower, asparagus, a green bean, maybe some broccoli or lettuce. You could also do mushrooms, carrots, peppers, maybe some berries, okay? So make sure that you have some type of produce. You don't need a lot. You need about a 1 centimeter square to put in your Petri dish. So just a little bit. Right? Now we're looking at carbohydrates. Carbohydrates can be in a monosaccharide form, an oligosaccharide, disaccharide, or it can get into the polymer form, which are the polysaccharides, remember? So we don't always need to have a polymer of carbohydrates, but certainly our body can process those and can make those. Now the simplest form is just going to be one monomer or a monosaccharide. This would be like glucose or fructose. Fructose is the sugar found in fruit, right? Now you can see different pictures of glucose and fructose on your lab procedure. And you can see fructose is actually a pentose or a 5-carbon sugar, so slightly different shape than our glucose. They can't be broken down into any smaller sugars. Certainly we could break apart glucose or fructose, but it wouldn't be breaking it down into a smaller sugar. That is the simplest form already. Fructose actually tends to taste a little bit sweeter than glucose for the same amount. And of course, when you look at these images, remember that at each intersection, that's a carbon atom with hydrogens coming off of it to make to make a total of 4 bonds. Chemists just don't always need to draw those out. They have a shorthand for it. We can then make a disaccharide, which would be 2 monomers coming together. Disaccharides are things like sucrose, table sugar, which is a combination of glucose and fructose bonding together. Okay, so not a polymer yet, just 2 monomers coming together to make a disaccharide. You could also combine glucose and galactose, which makes lactose, the sugar that's found in milk. So you should know some of these basic carbohydrates that we talk about and what they're made of. Sucrose, lactose. You should be able to recognize fructose and maltose. Okay. A series of glucose molecules, 300 to 1,000 maybe. They can get very large. This is polysaccharide. This is going to be our polymer form of a carbohydrate. And when we have all of these glucose molecules bonded together, we can make things like starch or cellulose or even glycogen in the body. Now for this experiment, we're going to be looking at starch specifically and the presence of starch in different food. So iodine, that deep reddish brown stain that you guys have in your lab kit, iodine can actually tell you if starch is present in a food or not, which is pretty neat. So it actually adheres to an amylose molecule, or a molecule that's in starch, to put it simply, and it will change colors, all right? So iodine does stain. It'll stain your skin and your clothes, so please be careful with it. And make sure that you look up the SDS sheet online for iodine before you get started, so you know what to do in case you spill it, get it in your eye, or anything like that. All right, now certainly you're gonna have your safety goggles. You're gonna be nice and careful for this. Now it says, iodine is a reagent that changes color in the presence of starch, and we're gonna use that iodine to determine if various food items have starch or not. Some of the items are food, some might not be, like our paper or paper towel. It turns out that carbohydrates can be used for a lot of different things, right? They're not just an energy source. That's what we use them for in animals, in the human body. But think about a tree, right? A big tree trunk is made out of a lot of different carbohydrates to give it structure, to give it support. So you're going to test each item in a petri dish. If you have to use the same petri dish, make sure that you wash it in between each sample. Or you can use a different petri dish for each sample, and that's probably the best way to do it if you have enough. And you just need a few drops of iodine for each sample. Okay, so go ahead and make sure you pause the video, you get everything assembled, and you read that SDS sheet for your iodine before we get started. Okay, so once you have your 4 petri dishes, you're going to get your 4 food items, and you want to put just a little bit in each petri dish. They say about 1 centimeter cube or square, so something just about the size of that. Okay, I have a blueberry, some Rice Krispie cereal, cucumber, and a paper towel. So that's going to be my paper product. You can pick 2 food items. Remember our categories above, so you want one to be in more of the grain category and one to be in the more of the fruit and vegetable category. And then you can pick whatever you want for your 4th item, anything you're curious about, whether it's a food item or not. So go ahead and put those 4 petri dishes out if you have all 4. And again, you want your iodine and a little pipette dropper. Now again, be careful not to let this stain. Maybe you wear an old t-shirt or an apron. And make sure to put on your safety goggles and tie your hair back, take off any long earrings or anything. Now you really just need 1 drop of iodine on each. You could do 2 drops, but I wouldn't do much more than that. And if you have items like my Rice Krispies or pasta or rice, you want to go ahead and crush them and break them up a little bit. So you can use a spoon just to get them into a little bit more of a powder. This just helps your iodine actually get in there enough, right? Thinking about surface area and the contact that it has. So once you crush it, make sure you kind of pile it all up back in the middle. My blueberry, I kind of just squirted out the insides. I'm going to crush it a little bit. Okay. And then for my cucumber, I actually just diced it up into some pretty small pieces. So however you want to do it, you want to just get some small bits in there of whatever you're testing. Now with your iodine, you're going to be putting a drop on all 4 of your samples, and you're going to be timing it for 4 minutes. You can do 1 sample at a time and wait 4 minutes, or you can do all 4 of them together. If you do all 4 together, just make sure that you're looking at all of them and you're recording it as accurately as you can at each time mark, okay? So you're going to be filling out that table, and you're going to be looking at time 0, time 2, and time 4. So time 0 is right when you put that first drop on it. 2 minutes later, you want to note again what it looks like, and 4 minutes, you want to note what it looks like. So if that's a little bit too much to do with all 4, just do 1 at a time, and that's perfectly fine. Might be nice to have a paper towel or something down just so you don't stain your countertop either. And you're going to pull some up into your pipette. You just squeeze that top bulb, okay? And then you're going to give it a gentle squeeze to a drop. I'm just going to do 1 drop onto all of my items. There we go. This would be time 0. I'm going to record what they look like at this point. I'm going to set my stopwatch, and then I'd come back in 2 minutes and record what it looks like then. Now you're looking for a color change to tell that starch is present. So your color to start with, of course, is that deep reddish brown. And so once you see your samples, you'll notice some of them might turn an interesting color, if you can see that. Oh, that's what I was trying not to do. Okay. Here's my cucumber. See right here. Paper towel. Looks kind of like blood. And then my blueberry. There it is. So pick some other types of things. Maybe you do a little overlap with what I do, but try to pick some different items that you have in your house. Put your drops on, and then make sure to wait all the way through 4 minutes to record if the color changed in any bit. Now, if the color changed with time 0, right when you put that drop on, probably means that you have a lot of starch present. So my Rice Krispies, I have this really deep blue navy kind of color. So that's showing me that I do have a decent amount of starch present in that Rice Krispie cereal. Sometimes it might take a little bit though. And so if you end up getting a color change after a few minutes, you do have starch present, just maybe a little bit less. Once you finish, make sure that you dispose of all of your samples in the trash. Okay, you want to make sure that you don't eat anything with iodine. So clean up your station as best you can. Clean off your Petri dishes and save them for next time. Same with your dripper as well. You can just put this under running water and squeeze and release this bulb to rinse out the inside. And with your iodine, of course, make sure you have that cap on nice and securely so that it doesn't accidentally spill. Okay, so for this next part, we're going to be talking about lipids. So remember, lipids are one of the macromolecules, but they do not form polymers because they don't have a repeating subunit. So they're typically very long carbon and hydrogen chains, maybe attached to something to make it a triglycerol, okay? Maybe it's more of a cyclical shape to be a steroid or a cholesterol. Whatever it might be, remember there is no repeating monomer though. So this is our one macromolecule that is not a polymer, even though they can be larger molecules. So for this, you're going to need a few different things. You're going to want to have your tightly sealed container that was included in your lab kit, your marble. You'll also want your 2 test tubes, and then you'll need your ruler, some type of marker, or a wax pencil if you have that, pipette, and then some type of vegetable oil. Okay, canola is fine, olive oil, anything like that, peanut oil. If you use coconut oil, just make sure it's warm enough to be in the liquid form. That one might be a little bit harder though. And then you'll need some dish soap, right? Some liquid dish soap. So for part C, it says lipids are generally hydrophobic, meaning that they are water-hating, right? It's because of the nonpolar bonds that they have. So they have these very long nonpolar tails. Think about our phospholipids. Most lipids are nonpolar, but of course we've seen some, like the phospholipids, that have a polar end to it. That's usually because of oxygen. Remember, oxygen has a very high electronegativity, so it pulls those electrons to it, causing a bit of polarity. So if we have a lot of those hydroxyl groups or OH groups, then we end up with a slightly polar molecule. Now, most lipids, again, are nonpolar because of those nice long carbon-hydrogen chains. Hydroxyl groups are, of course, the oxygen and the hydrogen that we talked about in the functional group chapter. And so looking down at the procedure, we're going to look at how lipids react in a couple of different ways. So this is where you need your test tubes. It says you're going to need 2 test tubes. And for this first part, you actually want to take your ruler, looking at the centimeter side, and you want to put a line at 1.5 centimeters and a line at 3 centimeters on each of your test tube. So in a lab, we would use a wax pencil because we could wipe it off. But maybe at home you use a Sharpie or something like that that'll stay on it, and that's fine. So at 1.5 centimeters, draw a line, and at 3 centimeters, draw a line. And you're going to do that with both of your test tubes. You're going to label one test tube number 1, and the other test tube you can label number 2. Right? Just like that. Now in the first test tube number 1, you're going to add your vegetable oil up to your first mark. So whatever vegetable oil you're using, go up to that very first line, and then you're going to fill it up to the second line with water. Now you can have your caps around. They're not the tightest. So I actually have a little container that I can put them in so my test tubes don't spill out. So maybe you want something like that. I'm going to use canola oil for this first part. So test tube number 1, I'm going to put oil in just to that very first line. And you can use a pipette for this, or maybe you just go very slowly. Okay, a little above, but there we go. And then we're going to fill it up to that next line with water. And we're going to do the same thing for both of these test tubes. So while I have my oil out, I'm going to go ahead and pour slowly up to my second line. There we go, that was a little better. And we can just use tap water. Just put it on very slowly so you don't overfill.
Good sign. How many little fox cover the house?
Okay. So it should look something like this. Right? And again, you can cap them, you can put them in a container and just kind of let them sit and do their thing. Okay. Now the first test tube we're going to leave just with water and oil. It says in the second test tube, we set it up the exact same way, but we're actually going to add 5 drops of dish detergent to that test tube. All right, so find test tube number 2, and you're going to add your 5 drops of dish detergent. So you can, if you have a dropper on yours, squeeze it out that way, but mine's going to run out a little bit too quickly, so I recommend using the pipette. So you want to go ahead and suck up some soap in your pipette and then add it to test tube number 2 and only to the second test tube. Okay, I've gone ahead and I've added my 5 drops of detergent to test tube number 2, and test tube number 1 just has my oil and water. So your tubes will probably look something like this. You want to record your observations in that very first table. Now if you went a little bit off from your lines like I did on this one, that's okay. Just make sure that they're roughly similar, okay? If we were doing this in a lab, we would be measuring everything out very exactly. There are actually pipettes that measure out how much you're sucking up into it and then pushing out. But at our home lab, it's okay just to be a little bit off. So write down what you see. What's on top? What's on the bottom? Do you see any interactions between the layers? Any qualitative observations, all right? Now, number 3, it says you want to mix the test tubes using a finger push. I'll show you what this is. When we stir test tubes, we never just want to swirl it around. Okay, that's not really going to do a whole lot. And you never want to put your thumb on top and shake it because we never know what's in the test tube, right? We could be using something very dangerous. We don't want to stick our fingers on top of it. So what we do instead is you want to have a nice firm hold on the top part of the test tube. And then you take your other hand and you really just kind of flick like that. You want to do it a few times. And we call this the finger push method or the finger flick method. And it actually helps to stir up the solution from the bottom. You can't really overmix this too much right now. So play around with it. Kind of think of a cat playing with a toy, all right? And then think about what you see. Did it change at all? And you're going to do that for both of your test tubes. So your nice finger flicks, do about the same amount of times as you do for each one. And of course, make sure you're holding that top end so you don't flick it across your room, right? Okay. So take some time to observe your individual test tubes. What do you notice? That's going to be your table under number 3. Now, number 4, it says lipids are not readily soluble in water. They don't dissolve in water, say like sugar does, okay? Think of oil and vinegar salad dressings. They separate pretty easily. Soap is a little bit different. We call it amphiphilic. Now, you probably remember this from our cell membrane chapter where we talked about phospholipids. So phospholipids are amphiphilic, and soap has those phospholipid molecules that give it the phospholipid properties. So remember, amphiphilic, we can think of amphibians who love both the water and the land. So amphiphilic means that they're both water loving and water hating, okay? They have a non-polar end and a polar end to it. Now, of course, we've talked about this with the cell membrane and forming those bilayers where the heads are oriented out towards the water and the tails are facing each other away from the water, nice and protected. We also mentioned something briefly called a micelle, all right? Now, a micelle is what you see on your picture right here. All of those white circular heads are facing outwards towards the water. The inside has the non-polar tails. And remember, this is what happens when we wash our hands with soap and why it strips our skin of oils, dries your hands out. Or maybe if you got oil on your hands after cooking, you use soap to wash it off. If you were just to rinse your hands underwater, it'd be really hard to get that oil off of your hands. But if you use soap, the soap actually forms a micelle. Your soap has amphiphilic properties, meaning it can surround those fat particles and wash them off with your water in your sink, all right? So, means that soap is able to put one of its ends onto the lipid and the other end out in the water, connecting the lipid and the water to help wash it away. So it says which side of the soap will face the lipid. Is it the hydrophobic or the hydrophilic side? Which side will face the water? Now take a look at the picture of the micelle. It says the micelle is formed when soap surrounds the fat or the oil, and it makes the lipid seem to disappear into the water, which wouldn't happen on its own, right? This micelle helps soap remove particles that would not have dissolved in water. The hydrophilic heads are facing out towards the water, and the hydrophobic tails are facing in towards the lipid. Bile acts in a similar way. If you've ever heard of bile produced by your gallbladder, part of your digestive system, it helps us break apart or emulsify fats that we eat. So bile acts as a surfactant to emulsify fats or break them apart in our digestive process. You can see the image over on the right-hand side. Again, it's just a representation, not what it actually looks like. But you can notice that the hydrophobic side of the bile faces the lipid, and that the hydrophilic side faces towards the water, right? Hydrophilic, water loving. This forms a micelle in the very same way that soap does, and it helps us digest those lipids that would normally not dissolve in the water in our body. Remember, we have a lot of water in our body. Bile tends to be more of a dark green or yellowish type of fluid, and that's actually produced by the liver, and then it's stored in the gallbladder, okay? If anyone's ever had their gallbladder taken out or know anyone who does, we can still function without it. You actually form a little bit of a pouch to store your bile where the gallbladder used to be. Pretty neat. Going down to number 5. We're going to go into a slightly different experiment now involving whipping cream, okay? So once you finish up with your test tubes and you've recorded your different observations on what they look like, you can go ahead and clean that up. You want to make sure to rinse out your test tubes really well, save them for any future uses. If you used your pipette, again, make sure to suck water up into it quite a few times to rinse out the inside so that it's usable again in the future. And we're finished with our oil and our soap, so you can go ahead and put that away. Okay, now for this next part, we're going to look at a little bit more of emulsification. Emulsification is essentially taking large lipid molecules and breaking them down into smaller lipid molecules. All right, why would we want to do that? Well, thinking about the bile in our above example, it helps us break down fat, right? And when we think about our metabolism chapter, metabolism in our body is all about those catabolic reactions that break things down so that we can use anabolic reactions to build back up what we need. So maybe I ingest olive oil, and that's the form of lipid that I take in, but my body really needs to build some cell membranes or some cholesterol or steroids. It can emulsify those fats, it can break them apart to help build the fats that I do need in my body. So emulsification is incredibly important. It's a catabolic reaction where we break down some of the larger lipid molecules, all right? Now, we can certainly do this with chemicals. We can also do this with some physical manipulation, which we're going to see in just a minute. If you ever heard of an emulsion is really a colloid mixture. Remember, colloid, we're thinking about the size of the particles. They're about that halfway size, okay? Not as small as a solution, not as big as a suspension, somewhere in the middle. But colloids are stable in that they remain mixed without us having to shake them, all right? So emulsions, once we go through the emulsification process with a lipid, we form an emulsion, and that emulsion is stable. It's a colloid. It will stay mixed. So some examples of common emulsions, mayonnaise, right? Egg yolks. What about butter, right? We took something that was fatty and liquid, and we made it into a colloid emulsification solution that stays constantly mixed, right? You don't have to shake your mayonnaise, it doesn't separate. So we're gonna look at that with some heavy whipping cream. So for this next part, you actually do need heavy whipping cream. You just need a very small amount. I think the smallest they sell it is just 8 ounces, but maybe you borrow it from someone, take a little bit from someone, you're not gonna give it back, of course. But you really just need a very small amount. And make sure you do use heavy whipping cream. If you use half and half or whole milk or anything else, it's not gonna work out very well. You want a tightly sealed container, okay? You probably got something like this in your lab kit. If you don't have one, just make sure that it seals very tightly. And you might wanna do this around the kitchen area, not near a laptop or computer, just in case anything leaks out. You also want your marble, okay? So it says for number 5, we're going to take a look at some lipids and the emulsification process. Obtain your plastic container and a marble. Smaller container means you're not going to have to work for quite as long, so a smaller container works pretty well. You want to fill up your container about halfway with heavy whipping cream. Place your marble inside the container with the whipping cream and then secure your lid nice and tightly, okay? Now, I'm not going to show you this one because I want you to do it on your own. Some of you might have done this. We do this every year on Thanksgiving, not with a marble, but with a hand mixer because it's a lot faster. But try it out with your marble. And what you're going to do is you're going to shake your container vigorously with one hand on the lid and one hand on the bottom until it's difficult to hear the marble anymore, okay? So once you don't really hear that marble as you're shaking it, then you can stop and take a look. It'll take probably 5 to 10 minutes. I'd guess closer to the 10-minute, maybe even 15-minute side if you have a really big container. So what it looks like, again, you will have heavy whipping cream halfway up in your container. I do not. But you want to have one hand on the top, part of your hand on the top, part of your hand on the bottom, just so that it doesn't open up and spill. And you're going to shake it vigorously for about 10 minutes. All right, you'll get a good arm workout in. You want to make sure that you hear the marble to begin with, and then by the end, as you're shaking, you don't really hear it anymore. At that point, you know you're probably at your end. It says you can look periodically as you wish. Hopefully you have a clear container and you can eyeball to see what's happening with it. All right, if you don't know what's happening, you'll learn for a nice surprise, I suppose. So it says, what state of matter is the cream before you shake it? So when you first put it into your cup, what's the state of matter? Is it a solid, a liquid, a gas, or a plasma? Okay. And then it says, what state of matter is the cream after you shook it up? So after you did all the shaking, what's the state of matter? Now, a lot of students kind of struggle with that. And my suggestion is to think about the properties of the different states. If something's a liquid, remember it tends to fill the shape of its container very easily. It will ooze into a certain shape. If you were to pour it on a counter, it would spread out across the whole counter and spill, right? Whereas a solid tends to hold its shape very well. So maybe you get a spoon and you scoop some out and you see what happens. Does it run off? Does it stay in a glob? Is it a solid or a liquid? Is it both? And below it says cream is an oil in water emulsion, all right? So cream, of course, comes from a cow. It's the milk that's produced, and the cow milk has a lot of different components, again, to raise its young. So thinking back to that chicken egg, that's really forming the basis and the nutrients for a baby chick. This is doing the same thing for that cow who's nursing from its mother. So milk has a whole lot of protein, it has a lot of water, and then it has a really nice, rich fat as well, just to help give that cow all the nutrients it needs as it grows. And of course, it's delicious for those people that eat it. So it's an oil in water emulsion, a colloid mixture, where the milk proteins help to emulsify the oil. What does that mean? The milk proteins are helping to break down those lipids. They're breaking it down into smaller particles. So when you shake it up, you're actually bringing that fat out of the emulsion and you're separating it, almost as if you were churning butter. Okay, so if you kept going, that's really how you make butter. Maybe you've made ice cream, maybe you've made whipping cream like this before. Okay, you're emulsifying those fats. And number 6, it says one of the most important roles that lipids have is the forming the majority of the cell membranes. All right. So looking at your test tubes, oh, hopefully you didn't clean them up. I told you guys a little bit too early to do that. If you didn't clean them up, you want to look at test tube number 1. You want to pretend that the oil layer is your cell membrane, which is made of phospholipids, right, on the outside of the cell. And the water is at the bottom of test tube number 1. It's inside of the cell, and it's that aqueous, watery environment. So take some food coloring and put a drop inside the test tube, all right? If you already cleaned them out like I told you to, my apologies, I got a little ahead of myself. Just make up another test tube solution. It's pretty simple, right? You just want to fill up your oil, put your water on top. That's all test tube number 1 is, no soap in it, all right? So again, sorry if I kind of jumped the gun on that. So for test tube number 1, if you have it already, if you need to make it up again, you want your oil, then you want to fill it up with your water, half and half, and then put some food coloring in there. Whatever food coloring you have in your lab kit. You really just need a drop, okay? It should sit on top of the oil for a little bit, and then you might notice that it drops down into the water portion. So think about that for a minute. Did the food coloring disperse or dissolve into the oil? Yes or no? Did the food coloring disperse or dissolve into the water? Yes or no? So what do you think? Is the food coloring polar or nonpolar? Right? We can tell if it's polar or nonpolar based on if it's dissolving in the polar or the nonpolar section of our test tube. Okay. Now, number 7, it says lipids are mostly made of carbon and hydrogen, and they're generally not soluble in water. Some common categories of lipids are fats, oils, waxes, steroids. Many lipids are formed when a glycerol molecule combines with 3 fatty acids. We call that a triglyceride, right? And it says, as you can see from the following diagram, a lipid does not have a molecular repeating subunit. There's no monomer, so it is not a polymer. Even though it is a macromolecule, it can be a large molecule, it doesn't have that repeating monomer. So in the diagram, most of the carbon atoms are, of course, just shown through the intersection of the lines. They're not actually written out. And we're looking at the difference between saturated and unsaturated lipids again. So think about way back in our covalent bonding lab, we had saturated versus unsaturated alkanes, alkenes, and alkynes, all based on if there was a single, double, or triple bond between the carbon atoms. That's how we label the lipids as well. So saturated lipids all have single carbon-to-carbon bonds, whereas unsaturated ones have a double or a triple bond somewhere between carbon molecules. So saturated lipids, because they only have single bonds, they are saturated with hydrogen atoms, right? They have as many hydrogen atoms as they could possibly have. Remember that every single carbon only wants 4 bonds coming off of it. Each line is representing a shared pair of electrons, so 4 lines really means 8 total electrons to fulfill the octet rule. So looking at this image on your lab report, is the lipid saturated or unsaturated? What do you think? course, this is your triglyceride, so we see those 3 fatty acid chains, and then that vertical glycerol molecule. And then you have a little note at the bottom. So our last macromolecule that we didn't really explore in this lab are nucleic acids. Now, nucleic acids are, of course, our DNA and our RNA. So they're incredibly important to the body, right? Now it says carbohydrates, proteins, lipids, and nucleic acids are the 4 macromolecules or biomolecules, life molecules that we talk about. Nucleic acids were first found in the nucleus, which explains the name, nucleic acid. However, DNA is too large to leave the nucleus. It can't go out, so it's always in the nucleus, right? RNA, on the other hand, is much smaller, and so it's able to leave through the nuclear pores. It's able to go out into the cell, into the cytosol, and that's where we actually synthesize proteins. So DNA stores the information that's telling the ribosomes how to make a protein, but we need that RNA to be able to be the messenger, to bring that message out to where the ribosomes are, okay? Ribosomes are, of course, the factory for producing proteins, if you remember your organelle chapter. In the ribosomes is where we take the RNA and we translate it into our amino acid polypeptide chain, and then it's folded into a functional protein. So DNA stores the information on how to make a protein, and the proteins make up most of the structures and the functions of the body. So nucleic acids are those really important 4th macromolecule, and we're going to look at it in future labs. Okay. That's it for this lab. Of course, ask me any questions as you go, and I hope you have fun.